Welcome to Modeling Embryonic Development, starring Typical Frog Family. We will be following frog development from fertilization to gastrulation. Here we have a lone sperm searching for an egg to fertilize. Fertilization in frogs is external, and the eggs are protected by a jellycoat that is released by the female. When the sperm makes contact with the egg, an acrosome, also known as a vesicle, in the sperm is released and breaks down the jelly coat. A sperm nucleus enters the egg cytoplasm, and fusion causes a fast block depolarization that creates a fertilization envelope, which prevents the other sperm from fertilizing that egg due to the formation of a protective layer. The binding and fusion of sperm causes the egg to activate. The sperm nucleus merges with the egg nucleus and creates a diploid nucleus, and DNA synthesis begins, and first cell division occurs.
Next is the four-cell stage. Remnants of the mitotic spindle can be seen between the two pairs of cells that have just completed the second cleavage division. During this stage, cells are totipotent and each can become a new organism. Accidental splitting of zygotes can create twins.
In the early blastula stage, after cleavage divisions, the embryo is a multicellular ball that is surrounded by the fertilization envelope, and the blastocele begins to form in the middle. In the later blastula stage, a single layer of cells surrounds a larger blastocele, and the fertilization envelope is still present. The embryo will hatch from it and begin swimming. The embryonic stem cells, also known as germ cells, form as the cell continues to split and eventually differentiate.
By gastrulation, primary germ layers form. All organs and tissues develop from these three layers. Primitive gut tubes forms. Gastrulation begins with the blastopore, which is green, which appears on the dorsal side of the late blastula, pink. The blastocele is shown blue. The crease results from cells changing shape and pushing inwards. Outer cells then move into the interior, where they will form the endoderm and the mesoderm. While this is happening, cells at the animal pole change shape and begin spreading over the outer surface.
The blastopore extends around both sides of the embryo as more cells invaginate. When the ends finally meet on the other side, the blastopore forms a circle that is smaller as the ectoderm spreads downward over the surface. Continued involution expands the endoderm and mesoderm and creates the primitive gut tube as the blastocele becomes smaller. The endoderm-lined primitive gut tube has replaced the blastocele and the three germ layers are in place. The blastophores surround a yolk plug.
Now, our frog is alive. Thank you for watching. The end.